I'm oral historian Mike Chappell. Today, June 24th, 2012, I'm interviewing Dr. David Orth for the Endocrine Society at its annual meeting being held this year at the George R. Brown Convention Center in Houston, Texas. Dr. Orth, would you please tell me a little bit about your family background?
Well, for myself, I was born on March 5th, 1933, which happened to be the day that Roosevelt closed the banks. I was born in East Orange, New Jersey. My father was a guy who had gone to Brown University. He had planned on going into advertising, but he came out of university during the Depression, and that wasn't a good time for advertising, so he ended up being an insurance man. My mom went to what they called normal school in those days, but it actually was a two-year teacher's college. And then she went back after my sister and I left for college. She went back to college, got her BA degree, and then took courses, graduate courses to maintaining her teaching certificate in Los Angeles. My grandparents, I didn't know either one of my grandmothers. They had died before I got to know them. My paternal grandfather went to the 8th grade, taught himself to be a draftsman and then an architect, and was reasonably successful all of his life. And my paternal grandfather was a store manager for Lowe Brothers Paints.
What type of early education did you have?
I went to 4 different public schools during the course of my early life. The one that I ended up with for high school was East Grand Rapids High School in East Grand Rapids, Michigan. And although it was a public school, it was basically a private academy because it was located in the wealthiest part of town, and the parents were very involved in the school and very willing to pay the property taxes that maintained the school. So it was, in terms of facilities and in terms of the teachers, it was, as I say, basically a private school.
What were your favorite hobbies and/or extracurricular activities?
Well, I always seemed to be messing around in nature. The places we lived, curiously, were both dead-end streets, and they ended in the woods. So I was always turning over logs and looking under rocks, and there was a nice swamp nearby, and so I was always collecting various things, which my mother didn't appreciate. But I would have pollywogs that grew into frogs, and I got caterpillars of various kinds and raised them and watched them go to through the stage of chrysalis development and or cocoon and form butterflies or moths. We've raised dozens of guinea pigs at one point. And then in the end, in junior high school, I got interested in tropical fish and ended up with several thousand tropical fish in dozens of aquariums down in our cellar, which my dad and I had built from angle iron and plate glass, some of them 100 gallons and more, and tried to breed them to get interesting colors in some of them. I was also interested in art. Art seems to run in the Y chromosome in my family. My grandfather, as I said, was a draftsman. My father was an excellent artist, although he never did it professionally. My son was a professional artist who went to Rhode Island School of Design and was a successful fashion illustrator in New York. And although I never had any formal training, my dad actually taught me how to use oil paints. I was active in art. I won a citywide competition in Grand Rapids with an oil painting I did as, I guess, a sophomore in high school. And I was art editor of the senior yearbook. So that's an interest I've maintained all my life.
Did any of your teachers at East Grand Rapids High School have a particularly strong influence on you?
Well, they were all great teachers, in fact, but one of them was especially important to me. Her name was Marie Yonkman, and she taught senior English. And Marie was a taskmaster. She had us writing endless papers all our senior year. She drilled grammar and sentence and paragraph construction and how to organize our thoughts to write a paper till there was no way we could possibly forget them. And she instilled in all of us, I think, a great love of English literature and of language. And she clearly influenced me more than anyone else for the rest of my life. Marvelous woman.
What drew you to medicine?
Well, I think it was a combination of my interest in nature and my bent for science. My father says that he wanted to be a physician, but he worked his way through college, which was not easy to do, and then he just didn't have the resources to go on to medical school. My mother told me that she told her mother, who died of breast cancer on her deathbed, basically, that her son was going to cure cancer. Those were burdens I didn't take too seriously, but I'm sure at some subconscious level they may have influenced my decision.
And why did you choose to attend Brown?
I had 4 criteria for a college. It had to be small, it had to be far away from East Grand Rapids, it had to have a good science department, and it had to be affordable. So I visited 4 schools. University of Michigan, because it was the cheapest for me and most affordable, but it fulfilled neither the requirement for being small or being far away. I went to Carleton College in Wisconsin because some East Grand Rapids graduates who had been there were very, very complimentary about it. I went to the University of Rochester in Rochester, New York, because it had a really good chemistry department. And I went to Brown University, which arguably had the best chemistry department in the country at that time, and was also my father's alma mater. And of the 4, I really liked Brown instantly, and just liked the feel of the place and where it was located in Providence. And furthermore, the man that I had interviewed at University of Michigan, a guy named J.P. Adams, had been a professor of math at Brown when my father went there. And J.P. was quite amazing and knew all about my father, 27 years after he'd last seen him. And J.P. said he'd love to have me at University of Michigan, but that I really ought to go to Brown, so there didn't seem to be much doubt about where I'd go.
While you were at Brown, how did a medical career figure into your plans?
Well, for a variety of reasons, it didn't. I went to Brown on a regular NROTC scholarship, and with that scholarship, you got full tuition, room and board, all your books paid for, 2 round trips from home to the college each year. And you took some summer cruises. The problem was that the Navy did not want physicians at that point. They wanted line officers, officers who were going to run the ships, basically. And so they didn't allow you to take a pre-med course. I had really not thought seriously about taking a pre-med degree anyway, and thinking that biology was a less practical major than chemistry in the long run, I decided to take a Bachelor of Science in Chemistry. In retrospect, I probably should have taken a Bachelor of Arts in Chemistry, because by taking the Bachelor of Science path, I really had no time left over after chemistry courses, chemistry labs, naval science courses which were required, naval science drill, and all of the other requirements of the Bachelor of Science degree in mathematics, 5 courses in calculus, 2 courses in physics, and so forth. I really had no time to take much else. And by the time I had decided that I really wanted to have a a broader experience. That was in the second semester of my sophomore year. I had already taken so many chemistry courses and science courses I couldn't major in anything else anyway, so I just stayed with it. I learned what I did about liberal arts by listening to my fraternity brothers cram for their exams, and got some by osmosis, I guess.
So then after Brown, you went right into the military?
Yep, I got my diploma from Brown, and about 15 minutes later I was commissioned an ensign, and 4 days after that I was on my way to Norfolk.
Now I know you did an interview with Adolf Friedman, and you discussed an oral history. It was an oral history, not a video history, and you discussed somewhat of your military career, but could you tell me just a little bit about it now?
Well, I had a great time in the Navy. Enjoyed all of it, actually. I was assigned to heavy cruisers in the Atlantic. I sort of talked my way into the operations department on the cruiser, and a job called Combat Information Center, which is sort of the brains of the ship for the radar and navigation and so forth. And I became what's called an all-weather air controller. It's not like a federal air controller in commercial air. We controlled friendly aircraft and directed them to enemy aircraft and directed the intercepts that they made. And I enjoyed that, had a great time doing that. And then because of my expertise in that, the Navy was just commissioning a new ship, the USS Boston, which was a World War II heavy cruiser that they were refitting as a guided missile cruiser. And they needed something as targets for their guided missiles. So they sent me to learn how to control drones. And so I started with little tiny drones, not much bigger than model planes, and went from there to World War II prop planes, and then Korean War jets, and finally to the Navy's first cruise missile, the Regulus I, and got shot down, I think, something like 18 times and never had to parachute.
Now, did you commit to a career in medicine while you were in the military?
While I was in the military, two things happened. Number one was because I hadn't really had much in the way of liberal arts in college, I started taking correspondence courses in literature and writing and biology. And I also noticed that time was just going by really fast, and that committing to another 4 years of education wouldn't really be that big a deal. So about, I'd say halfway into my second year, I decided that I would go into medicine, and I had sold myself at the end of my college career, since I did not, I thought, ever want to be in a laboratory again, which turned out to be ironically where I did end up, but I sold myself as a liaison person because I had done a fair amount of art, I'd done some writing, and I had the scientific background. I presented myself as someone who could be a liaison between the scientific aspects of a company and their advertising agencies. And they liked that, and so I got nice offers from General Mills and from Procter & Gamble. And Procter & Gamble offered actually to give me a stipend while I was in the Navy, which I'm glad I didn't do. And so when I decided to go to medical school, I wrote Tom White, who was the guy who had recruited me, and told him what I decided to do and thanked him, and applied to the University of Michigan, which was the school that would have been the least expensive for me to go to.
But you ended up in Vanderbilt. How did that happen?
Well, the last 6 months of my naval career, in about March or April of that time, having already been told that I was admitted to Michigan, the admissions dean there thought, in retrospect, that a course I'd taken at Brown from the biology department was actually a chemistry course. And so I didn't have enough biology to meet the requirements, and that meant that I couldn't enter the class until the year afterwards. So the question then was what to do. I was married. I needed to do something at that point. And my then father-in-law Randall Stewart was the chairman of the English department at Vanderbilt, and he said, well, why don't you come down and look at Vanderbilt? And I had a very provincial Yankee view of the South at that point. I had sort of this vision of the natives, shuffling up through the red dust in bare feet to the medical center, and was, of course, totally surprised when I got to Vanderbilt. Had a wonderful experience, loved the place immediately, and they put me on the waiting list for that fall. But unlike the previous 2 or 3 years, all the freshman class showed up, and so I had to wait for a year. And during that time, I took some more undergraduate courses to make me eligible for any medical school, and then worked for half a year before going to medical school.
How did you become interested in research, and what research did you do while you were in medical school?
Well, I'd always been interested in how things worked, even as a kid, and I think that's what probably got me into science and chemistry to begin with. And while I was taking physiology in my first year at Vanderbilt, Howard Morgan, who was an assistant professor in physiology, got me interested in the project they were doing. He had a system which was an isolated perfused rat heart, which is an actual beating heart in a kind of a test tube being perfused by solutions that duplicated plasma. And a pair of investigators from Yale, Barnett and Ball, had published just recently an article that proposed the idea that the way insulin worked was that it stimulated what's called pinocytosis. And pinocytosis is a process whereby the cell membrane pinches off a little sphere from the outside and carries it inside the cell. And they suggested that maybe insulin worked by stimulating pinocytosis, and it took the glucose in with the liquid that was in the pinocytotic vesicle. So we decided to test that hypothesis and look at a couple other situations. So using that system and the electron microscope and a whole lot of time during my summers and spare time in medical school, we proved basically that that was not the way that insulin worked, and published a paper. And so that kind of introduced me to medical research and piqued my curiosity.
And how did your choice of endocrinology as a subspecialty come about?
Well, I'm a firm believer that that life is just a series of accidents, and how your life turns out depends on how you respond to those accidents. And this accident was the fact that for my hospital medical rotation, my last name began with O. And so on the basis of that, 3 of us were assigned to what was then the sort of nascent, the beginnings of the Clinical Research Center at Vanderbilt. And on that service were 2 interns, Larry Wolf and Abba Caston. There was a resident, Philip Feltz. There were the 3 students, myself, Bob Northcutt, and Bud O'Connor. And the attending was Grant W. Little, who was probably one of the best endocrinologists and certainly one of the best clinical investigators in the world at that point. Bud O'Connor had already decided before he went to medical school that he was going to become an ophthalmologist, so I guess nothing could have dissuaded him, but the rest of us, unfortunately, or fortunately as it turns out, were under the spell of Grant Little, and everyone else on that service became an endocrinologist, so that's how it happened.
Could you say a little bit more about what Grant Little was like as a mentor and a leader?
Grant was a phenomenon. He was a legend in his own time, and I think that he enjoyed fostering that image. He had a commanding presence. He was referred to as the Great Stone Face by some people. He had a very, very square face with very powerful jaws and bushy eyebrows and deep-set eyes, and you knew you when you were in his presence. He had an incredible way of thinking through a problem that reminded me powerfully of my father, who really was an extremely logical guy. He had a way of taking a problem and stripping away all the incidental and unnecessary kind of parts of the problem and reducing it to a single central issue, which usually only had 2 or 3 answers. And Grant had a way of proposing often just a single experiment, the answer to which would tell you whether the answer was A or B. And he had such a remarkable intuition about endocrine physiology that he usually knew the answer before the experiment was done. He just had an enormous influence, I think, on almost anyone he came in contact with.
What kind of patients did you see when you were working with Grant?
Well, as a student, we saw practically every endocrine patient known to man, certainly all those that had abnormalities of the hypothalamic pituitary adrenal axis. In addition to that, taking care of those patients on the service, we also participated in the research rounds that Grant held every afternoon, so that we kind of saw the way his mind worked, how he absorbed the results and designed the experiments that would be performed on the patients for the next few days and so forth. We heard the fellows interact with him, and it was a pretty intense kind of an experience. So it was not strange at all that we ended up as endocrinologists, I think. He made endocrinology at that point in time seem to me, and I think to the others, as the only, without throwing off on anybody else, but as the only truly rational discipline in medicine. The systems were understandable. Many of the hormones, or at least we thought at that time, many of the hormones were known. You could dissect the normal and abnormal things that were going on with those systems, and the things that you didn't know, you had the feeling that with the proper experiment and with available tools, you could figure out what the answer was. So it was intellectually a very satisfying discipline, as he described it.
At the conclusion of your medical school education, how did you envision your career coming out?
Well, I had pretty much decided that I wanted to be in research, and by doing that, that meant that I pretty much would be in academics. I decided that I wanted to be in endocrinology. As I was thinking about that, one thing I did not want to do was to be primarily an administrator, although that also turned out not entirely to be the case. And I talked to David Rogers, who was then the department chair of medicine at Vanderbilt. When he was appointed, he was the youngest chair ever appointed, and was a very available sort of guy, and I had a good relationship with him. And I talked to him about my plans, and he reassured me that because of changes in the way that medicine was being funded since World War II, primarily through grants from the NIH, it was no longer necessary, and it was no longer an inevitable path that you went from being an investigator to a division chief to a department chair to a dean or whatever, that as long as you did good research and had good ideas and kept things going, you could do research as long as you wanted. And so that was reassuring to me.
And how did you come to do your internship and residency at Osler Medical Service, Johns Hopkins?
Well, I got urged to go to several places from people on the Vanderbilt faculty whom I admired, most of them clinical faculty, actually. So I applied to 4 places in order. I applied to Johns Hopkins first, Massachusetts General second, Columbia University third, and Vanderbilt fourth. And The interview at the Mass General was a humbling one. The three people who interviewed me were Holly Smith and the two valets. The valets were experts in calcium metabolism, which was my weakest point, and I had a very uncomfortable interview there. I was not accepted, as it turned out, at the Mass General, but I got a very, very nice letter from Walter Bauer, who was chairman of the Department of Medicine afterwards, which was, I appreciated very much. And I was matched with Hopkins, so I got my first choice and all was good. When I got my acceptance and told Grant that I was going there, he said, well, that's great. He said, that's good. He said, now, I'd like you to come back to Vanderbilt and do a postdoctoral fellowship with me. And he said, I'm not being altogether altruistic in that. He said, because I think that I can give you as good a postdoctoral training as any place in the world. But he said, if during the next 3 years you should find a place that you think has a better program, let me know and I'll take it over. And this is absolutely classic Grant Little. He wasn't kidding. I mean, that's just exactly what he would have done. So I went off to Hopkins with the idea that I would learn there how to take care of acutely and seriously ill patients. And I wanted to do that because I felt that I needed those skills to take care of patients in a clinical research setting, because patients do get sick and you need to be able to take care of whatever happens. And then I got exactly what I wanted there. It was a fabulous experience. I think that all of us at Hopkins learned more from each other than we did from the faculty. We had good, obviously wonderful attendings, many of them superb, but my 3 years there, I can't recall more than 1 or 2 times that an attending, no matter how good he or she was, came up with an idea or pointed out something that one of the interns and residents hadn't already thought of and addressed. The house officers were just a superb group of individuals and physicians, and I've kept in touch with some of them ever since.
When you finished up at Hopkins, then how did you knew you were going to go back to see, to work with Grant Little at Vanderbilt?
Well, yeah, I had decided I would go back, and I thought I would do a 2-year fellowship and get my training, and then I would go off to some other medical center somewhere and join a Department of Medicine as an endocrinologist and begin my career. And as it turns out, that's not the way it worked out. I never left Vanderbilt, never found a place that I wanted to be more. So,
would you outline some of what you consider the most important aspects of your career?
Well, I was one of these people who tried to do all things. I knew I wanted to take care of patients. I knew I wanted to do bench research. So, in terms of my career, it's hard to pinpoint any one thing. I'd say, first of all, that I really enjoyed taking care of patients. I think I was a very good physician, probably never a better general internist than the day I left Hopkins. I think probably I degenerated after that. But I became a very good endocrinologist and certainly very good at taking care of Cushing's patients, so that was rewarding. In terms of my teaching, I enjoyed teaching medical students, but the thing I got the greatest reward from was mentoring postdoctoral fellows. And that was always a two-way street. We had many postdoctoral fellows. I had many from around the world, lots of them from Japan. And they were all amazingly industrious and amazingly inquisitive and talented people. And I learned as much from them, I suppose, as they learned from me. I know that when I retired, I certainly missed that intellectual stimulation. The pleasure I got out of it was doing what my dad and Grant Little had done for me, and that was teaching them how to think about a problem, how to strip it down to its bare essentials, and how to think about a way in which you could resolve the basic issue one way or the other. I think I also enjoyed passing on what Marie Yonkman had passed on to me, and teaching them how to write. And medical students really don't need to know how to write very much when they come to medical school. They don't have that as a major criterion, and many of them had not had the good fortune of having somebody like Marie Yonkman in their backgrounds. And so I would always have the fellow who had worked on the research project write the first draft of the paper. And then they would, these were in the days before there were computers and so forth, so they were typed out, you know. And so I'd have them type it out double-spaced or triple-spaced and give it to me. And then I would get out my red pen and start going over it. And the fellows, some of them actually took these manuscripts with them as sort of mementos of their days with me, because they call the manuscripts being covered with rivers of red. I mean, I sometimes had so many corrections that I had to go onto the back of the page, the opposite side of the page, to get them done. But they learned how to write, and they learned how to think. And I think the third thing you teach them is the discipline of science. I told them that doing science is fun and easy. Writing science is tough and hard, and sometimes not so easy. But if you're not going to share the science, there's no point in doing it. That's what it's all about. So that was a great thing. My administrative time at Vanderbilt was more or less one that was forced upon me. I was not interested in being head of the division. I just wanted to be left alone. But Grant Little became department chair, and although he thought he could both run the division and run the department, that turned out not to be the case. So I was sort of the joint director of the division at that point, and I thought it would be good for someone else to be there, so David Rubin came. We recruited him. I met him at Hopkins and urged Grant to bring him on, and then David became ill shortly after he joined us, and he died of ALS, myotrophic lateral sclerosis, a bad disease. And so at that point, I decided that I'd just give it up and become head of division, and I enjoyed it. It was good. I enjoyed the successes of the faculty. Al Powers, who's one of the people that I hired for the division, is done just famously well at Vanderbilt and also in the society, and so I've really enjoyed those kind of things. But the administration per se, and pushing the papers and attending the committee meetings, I did not enjoy. And then the last thing I think that I consider important in my career actually was my time with the Endocrine Society. I got into that because of another friend, Bert O'Malley, who got me to join the finance committee, and through that I got into other offices, and I enjoyed everybody that I met in the society. I found there was no turf guarding. There were some pretty aggressive personalities and assertive personalities, but everybody basically was collegial and worked for the good of the society and the discipline, and I think I helped a little bit to ease the society through a major transition from a fairly small society with limited membership and limited scope in terms of what they thought the society to do, into one with a much broader vision that's now doing things that none of us could have dreamed of at the time. So that was also an enormous satisfaction.
I want to ask you a little bit more about the society later, but for now, when you returned to Vanderbilt and you started on your postdoctoral research, what did you begin with?
Well, Grant had a dictum, really, for postdoctoral fellows. He thought that they needed to do two kinds of things. One, a project that was pretty much a surefire thing, that no matter what you found, which answer to the question you found would be publishable. And then they should have a broader, riskier, maybe more long-range kind of project. And so I was at Vanderbilt. Grant's area was the HPA axis, particularly the adrenal, and so obviously my interests were going to be in that area. So there were two projects that we decided on, Grant and me. I think he suggested several others to me, particularly in the area of mineralocorticoids and hypertension and so forth, and I was not interested in those. The one that interested me for the sure thing was working on diurnal rhythms. The diurnal rhythms were important in shift workers and in pilots who flew transatlantic flights and so forth. We knew that the HPA axis was involved in the circadian rhythm. The cortisol was high in the morning, low in the evening, and so there was something we could measure to look at the circadian rhythm. And the other one that I was going to do, decided to do, was to work out a radioimmunoassay for ACTH. And the reason for that was Dr. Little was not an expert in peptides. There were no radioimmunoassays at Vanderbilt at that time. And in order to sort of have my own program, my own thing, I decided to do something different from what Grant was doing.
Regarding the circadian rhythms, then, your SAFER research program, what was the dominant paradigm at that time?
Well, that was peculiar because there had been a fair amount of work done in circadian rhythms in animals and plants, and in every situation it was shifts from darkness to light or light to darkness that were the what were called the synchronizers, or the Germans call them the Zeitgeber, or time giver. Circadian means about a day, and what that means is when you looked at these rhythms, it turned out in constant light or constant darkness that the rhythms had a period. They continued to go on, but they had a period slightly longer than 24 hours, usually about 24 and a half hours long. And they would continue to run forever. But as soon as you put daily light-dark cycles into the picture, they would then become 24 hours in length. And the way they did that was, if you were a diurnal animal, for example, as we are, when the shift from dark to light occurs in the morning, it would pull the rhythm back 30 minutes, basically. So it was a 24-hour rhythm. Then it would go on again, and if it didn't get the Zeitgeber the next day, it would gradually drift off until it was, again, 24 and a half hours long, and you'd have to get the Zeitgeber to bring it back. For some reason, the studies that were being done in humans were mainly being done by a group of Germans, led by an eccentric character named Franz Hallberg, who walked around in a kind of a Dracula-like cape, and a very interesting guy. And they had studied subjects in Lapland, in very far northern Scandinavia during the constant darkness of winter and the constant light of summer. Another one of these gentlemen had locked himself in a World War II air raid bunker and sealed him off completely from all light and dark cycles outside, and all social connections and so forth. And as a result of these studies, they had concluded that rather than light-dark cycles in man, that it was some sort of amorphous social schedule which controlled the light-dark, or would control the circadian rhythm. And that made absolutely no sense to me, and certainly no sense to Grant. So we decided to test that hypothesis, and we weren't interested in going into an underground bunker or traveling to the far north. And we had on the Clinical Research Center at that time a suite of rooms which consisted of a bedroom with 3 beds in it, a bathroom, and an entryway, basically a kind of a lock for infectious disease, but we decided to use it as a light lock. So we boarded up using actually rubberized sheets glued to the wall, pasted to the wall. We shut up all the windows and all the lights in all of those rooms. We removed all of those things like radios and so forth that might have light emanating from them. So the place was in total photographic darkness. And then what we would do is we could control the light-dark cycles from their eating schedules or anything else we wanted to do. So we would put people in there and put them on an altered schedule. Before we started the altered schedule, we'd do blood sampling every hour for 24 hours for cortisol, which we were going to use as our marker. And then after generally 3 weeks of having them in this environment, we'd sample them again and see what the effect was. So the first thing we tried to do was to see how much you could stretch the circadian rhythm. And we did days, in quotes, from 6 hours to 33 hours. And we found that you could certainly disrupt the circadian rhythm, but you could not stretch it very far away from 24 hours. And in retrospect, knowing how the thing works, that makes perfect sense. The next thing we tried to do was to find out whether the shift from dark to light was the important thing. And the way we did that was quite simple. We had our subjects in the dark room. They did everything. The schedule was exactly the same. They got up at the same time every day. They had their breakfast at the same time. They showered and all the rest, all in total darkness, because we simply prolonged the darkness for 4 hours from the usual time. So the lights went on at noon rather than going on at 8 o'clock. And 3 weeks later, their circadian rhythms had all shifted 4 hours later. So it was clearly the dark light cycle that was giving the signal, not their social schedule. And then we were interested in how that was perceived, because in some lower animals it was thought that the pineal body, which is, quotes, third eye in lizards, for example, and produces melatonin, which is a sleep-related hormone. So we wondered whether they were actually getting the light through their skull rather than through their eyes. And so we wanted to study blind people. We studied one blind man in one of these experiments, and we noticed that his cycle shifted during the 3 weeks, but it did not shift the same way everyone else's did. And when we then calculated what the length of the rhythm would be to get him from where he was at day 1 to 3 weeks later, and it turned out it was about 12.5 hours. That was not really the kind of proof we were looking at. So we were contacted by a lady. She contacted us, actually, having heard about us. Her name was Mildred Hamby. She's an ex-retired school teacher. She'd been blind all her life. Marvelous woman. And so we had Mildred on the CRC, and we put into her a double-barreled catheter that Andy France at Columbia had taught us how to make. And we infused through the outer lumen of the catheter a weak solution of heparin to anticoagulate the blood. And we withdrew through the very tiny inner catheter blood continuously from Mrs. Hamby for 51 days. And it was withdrawn into a vacutainer. We didn't use a peristaltic pump or anything. The vacuum was enough to draw it. And so we changed that vacutainer tube every hour for 51 days, and then we plotted out the data, and by a process, and a mathematical process called autocorrelation analysis, we showed that she had a free-running rhythm of 24.5 hours, basically. The interesting thing we discovered with Mildred was that she had cyclic insomnia, and she had that while we were studying her in the CRC, and it turns out that her insomnia, her difficulty in getting to sleep, was when her peak cortisol levels were occurring about the time when she was ready to go to sleep, rather than the normal time for all of the rest of us when we wake up.
How did this research advance the field?
Well, I think it put to rest the idea that humans are different from anybody else. Our circadian rhythms were governed in the same way as the rest of the plant and animal kingdoms. Secondly, our work certainly helped in looking at the issue of shift workers. so that nurses, for example, who were put on a night shift for a week or 10 days and then shifted to another shift after that, it's a terribly inefficient way to do things, because by the time they get acclimated to the new schedule, why, you shift them again and they become inefficient again until they acclimate. It became important for transatlantic flights. And so there were a lot of applications in terms of sleep and efficiency and so forth. And Robert, I think it's Charles Czeisler at Harvard has done, I think, the most work on this area. He now has a sleep institute at Harvard and has done a lot of innovative and very important work, I think, since that time. I got out of it after finishing those experiments.
Now, you said you had a risky option that you were going to pursue at the same time. That was Grant's idea that you should have both going on. What led you to choose the developing RIA?
Well, the RIA had just been developed by Solomon Burson and Rosalind Yellow. And that came about because Burson, who was a diabetologist, had observed that patients who got injections of insulin over a period of time, they had to have more and more insulin. And he discovered that the reason that they had to receive more and more insulin is because they developed antibodies to the insulin, which was foreign animal insulin. And the bound insulin was not biologically active, was not available to do its thing. And then, and to prove this, he had to prove that it was bound to antibody, which he did with Rosalind Yallow, who is a nuclear medicine person. And then it occurred to them, I don't know which one of the two, that that technique could be used to develop an assay. So what you would do would be to have an antibody, develop an antibody. You'd have a hormone you could track in some way, and in those days it was tracked by radio labeling it with 131 iodine. And you would then add increasing amounts of a sample or a standard, and they would compete for a limited number of binding sites with the label, and as the label came off and became free, you could calculate the ratio of the antibody bound to the free, construct a standard curve, and you could put an unknown in and calculate the bound over free ratio, and that would tell you exactly how much hormone was in the system. So that's where it was. The hormones that had been looked at that point were all peptide hormones, and they all were circulating in plasma at a fairly high concentration. It was rumored that Yallow and Burson were working on an ACTH radioimmunoassay, but there was no strong evidence for that. They hadn't published anything. And so the issue at that point was that the way that ACTH was measured was using a bioassay, which required a lot of blood, and the ACTH radioassay offered you the opportunity of measuring it as often as you wanted in very small quantities of blood.
Now, how did you proceed?
Well, I should, I suppose, tell you how the ACTH bioassay worked, because that was pretty remarkable. That was a modification of the Lipscomb-Nelson bioassay. You took a pint of blood from the patient. You extracted that by a long and laborious procedure. Then you took that extract and injected that into a rat who had been hypophysectomized, whose pituitary gland had been removed a few hours earlier. And you injected that in the femoral vein, and then at a set interval after that, you put a tiny little needle into the left adrenal vein of this animal and withdrew for a set period of time the blood that came from the left adrenal vein. And then by the amount of corticosterone, the glucocorticoid in rodents, that came out in the adrenal vein, you could determine how much ACTH you had injected. So it was a laborious, it was an expensive, it was slow, and it took so much blood that you could only measure a single ACTH in a single patient once every month, month and a half. So there was a huge, it seemed, reason for having an ACTH radioimmunoassay. So what we did was to start developing a radioimmunoassay, and for that you need basically 4 things. You've got to have an antibody that will bind ACTH, and it has to bind it with very high affinity. ACTH had the additional difficulty that it circulates in concentrations 1/10 or less of those of the peptide hormones that were being assayed by radioimmunoassay at that time. So it had to be a very sensitive radioimmunoassay. So you need that kind of antibody. Second, you need a labeled tracer that will, the antibody will bind to. Third, you need to have a purified standard so that you can construct a standard curve to compare your samples to. And fourth, you have to have a way of separating the bound from free hormone. And when I started this, the way you separated was a process called chromatoelectrophoresis. Chromatoelectrophoresis, the way it was done was you got a specific kind of filter paper from Whatman in England. You cut it into strips. You soak the strips in a phosphate buffer. You stretch these strips across an apparatus with a well of phosphate buffer at either end. And then you took a pipette and put the sample that you were interested in on the wet strip at one end of the wet strip with a dye that told you where the peptides were so you could follow how far they'd gone on the strip. And then you applied an electrical current to this apparatus with the anode on one side and the cathode on the other of these wells. And over a period of several hours, the peptides would migrate, the free peptides would migrate down the strip, and the bound peptides would stay sort of at the origin. And then this, by the way, is all done in a walk-in cold room. I got a lot of use out of my Navy pea jacket while I was working in the cold room for hours every day. Then after that was all done, you took the strips out, you hung them up on a wire and let them dry. Then you cut the excess strip off either end that you didn't need. You taped them end to end together with Scotch tape. Then you ran them through a gamma counter that had a graph output of the counts. So you got hills and valleys of radioactivity that appeared in this graph paper. And then you approximated the area under the peaks of free and bound for each one of these samples by triangulation. You sort of approximated it with a triangle and calculated 1/2 the height times the width. And that gave you a B over ref ratio. And then you were finished. And you know, that took a long time. It was a very laborious procedure, but that's the way it was done. And the problem with all of this was that you didn't know if you had a decent antibody unless you had a labeled hormone that you knew bound, and you couldn't know whether you had a hormone that bound unless you knew you had a good antibody. So it was a situation much like an old saw that my dad used to say. He would say, if we had some ham, we could have some ham and eggs if we had some eggs. And precisely the situation with the radioimmunoassay. A postdoctoral fellow at that time, Kaoru Abe, was doing an MSH bioassay that was just as laborious and just as difficult as the ACTH bioassay. And so he decided that he would develop a beta-MSH radioimmunoassay. So Abe, as he liked to be called, and I worked together, and Don Island, who was Grant's main man in the lab, did the radioiodonations. And we just started immunizing animals. So we immunized dozens of guinea pigs, rabbits, we immunized a goat, we immunized several sheep, we even immunized chickens, which is an interesting experience. And we just started testing these antibodies to see if they bound labeled ACTH. And in the meantime, Don Island was trying to learn how to label ACTH with a single iodine atom. And the reason for this is that an iodine atom is actually a very, very large atom. And when you put it on a small molecule of a peptide, if you get more than one iodine on, you run the risk of just the space that the atoms occupy, preventing the antibody from binding to the peptide. So we went on and on with this, and it took us about 2 years, but we finally developed the first 2 of dozens of antibodies, of radioimmunoassays that we developed over the years.
Now, he was working on MSH, and you were working on ACTH.
Correct.
What was the overlap? How did it come about that you both ended up working together then?
Well, we both wanted to develop radioimmunoassays, number one. Number two, the two were in strange ways connected to one another. They had similar structures. They were seen to be found in similar tissues in the pituitary and so forth. And so it was kind of a logical connection for us.
What issues arose concerning the physiological relevance of the RIA in general and your RIA in particular, and how were they resolved?
Well, we were fortunate in developing an assay for ACTH because the structure-function relationships of ACTH were very well known. They'd been worked out primarily by Siba Geigy in Switzerland, who had synthesized all these analogs and fragments of ACTH and MSH, looking for uses for pharmacological agents. So using these fragments and analogs, we tested them with our antibodies, and we could tell very clearly exactly where our antibody bound to the ACTH molecule. Because of Siba Geigy's work, it was known precisely where the biological activity of the molecule resided. So ACTH is 39 amino acids long. It was known that the majority of the biological activity resided in the first 18 amino acids, and it was known that if you did very much in terms of changing those first 18 amino acids, biological activity disappeared. So it was absolutely apparent that the chances of developing a radioimmunoassay that would measure precisely the biological activity of ACTH was very, very small. And that depending on where the antibody bound to the molecule, the ACTH could last in plasma either a shorter time, about the same time, or a longer time than the bioactive ACTH did. And we did experiments to prove that. We took individuals, we suppressed their endogenous ACTH secretion with dexamethasone, then we injected them with synthetic human ACTH, and we followed them every few minutes by sampling blood and assaying biologic ACTH and ACTH by various radioimmunoassays. We were able to do this, by the way, in the biological activity because we injected a huge amount of ACTH, so we didn't have to have huge amounts of blood. And exactly what we predicted happened, that is that the immunoreactivity disappeared more rapidly than the biological activity, and the immunoreactivity disappeared at different rates depending on what particular antibody you were using. This was anathema to Gallo and Burson, who were quite understandably trying to establish the physiological relevance of these peptides, and they objected quite seriously to our findings, but eventually this was found with other peptides. As a matter of fact, Rosalind published data herself, and it became perfectly obvious that this was true of all peptide radioimmunoassays.
How was your ACTH assay put to use?
Well, that turned out to be an interesting question at first. We unexpectedly faced that issue. Here, we had an ACTH radioimmunoassay, which was great, but we also had a very good cortisol assay, and you could measure cortisol in small amounts of blood. And cortisol was a very good marker for ACTH, so what was the point? And what we did was a number of things. First of all, we started measuring ACTH in tissues, and because of its sensitivity, we could measure it in tissues that didn't have much ACTH in them, or we could measure it in very small bits of tissue that had lots of ACTH in them. And so using the radioimmunoassay and looking at various species and various tissues, using the antibodies that we developed in collaboration with Bob Pfizer and Sam Spicer at South Carolina, we looked at the pituitaries of humans and other animals with both the ACTH and MSH assays, and we found that they were there together. And gradually we developed this theme that wherever we look for ACTH, whether we're looking at it in tissues in man or other animals, whether we're looking at its levels in humans, its disappearance from the plasma in humans, everywhere we looked we found that these peptides tended to track one after another. We also looked into the nature of immunoactive beta-MSH in man and found that it was not beta-MSH, but it was actually a larger molecule called beta-lipotropin, and other investigators at St. Bartholomew's in London published a paper showing exactly the same thing. So beta-MSH, as far as we know, does not normally exist in man.
Would you want to say more about how this assay advanced the field, or do you think that covers it?
No, I think that we were able, because we could assay ACTH so often, we learned much more in detail the dynamics of how ACTH was secreted. We had got a much better view of the circadian rhythm by comparing ACTH and cortisol levels throughout the day. We were able, as I mentioned, to show that ACTH was present in many tissues and many species, and that it always traveled with ACTH. So I think we moved ahead.
How about the clinical applications of it in terms of Cushing's syndrome?
Cushing syndrome is an interesting disease. It's one that occupied my interest for a whole 30 years. And with the ACTH assays and the MSH assays and later the beta endorphin assay, we were able to do a lot of interesting work with that. Cushing's, I think, is one of the most interesting endocrine diseases for a variety of reasons. Number one, it can be caused by pituitary tumors, corticotrope tumors. It can be caused by corticotrope hyperplasia. It can be caused by ectopic ACTH secreting tumors, tumors that are non-pituitary that make ACTH. It can be caused by ectopic CRH producing tumors, CRH being corticotropin releasing hormone. It can be caused by adrenal adenomas, that benign tumors of the adrenal, by malignant tumors of the adrenal. It can be caused by macronodular and micronodular hyperplasia of the adrenal, both of which can be either sporadic or, particularly in the case of micronodular, hereditary. It can be caused by physicians giving patients steroids knowingly or unknowingly. Sometimes they're contained in drugs that are not always apparent. One of my postdocs tells me that the major cause of Cushing's in Taiwan is traditional herbal medicines that the Chinese use, because the herbal medicine people stock them with prednisone, which makes you feel very good, but unfortunately also causes Cushing syndrome. So an interesting thing. And then in addition to that, the Cushing syndrome can be intermittent, it can be cyclical, and on top of all that, it's so rare that the average endocrinologist only sees 1 or 2 cases perhaps in his entire career, and so it remains a challenging kind of syndrome for endocrinologists to deal with. In our situation, we saw lots and lots of patients with Cushing syndrome, which was lucky for us. We were able to characterize in detail the nature of the disease. We saw patients who had unusual variations of Cushing syndrome, and were able to publish those and to explain their physiology and to elucidate how they fit into this overall picture. So I think without the ACTH assays, we wouldn't have been able to do that. And with the ACTH, MSH assays, and the ones we developed after that, they really formed the basis of all the things that I did in my lab for 30 years.
In what ways did the understanding, diagnosis, and treatment of Cushing's remain the same since that time, and what, in times of when you were actively researching it, and what ways has it changed?
Well, I think that a number of things have changed since I started, not since I finished my career, certainly since I started my career. The radiological procedures have advanced considerably. CT, MR, and gadolinium-enhanced MR, PET scans, and so forth have all enabled us to look at both pituitary and non-pituitary tumors much more easily than we did before. Diagnostically, I guess the single probably major advance was the development of inferior petrosal sinus sampling with stimulation by CRH. That has allowed us to confirm the diagnosis of Cushing's disease by seeing that ACTH is coming from the pituitary gland in these patients. And by using CRH to stimulate that secretion, you can frequently lateralize the tumor to one side or the other of the pituitary, which is of great assistance to the neurosurgeon with or without the imaging that was done prior to that time. In terms of the understanding of the disease, I think that we know much more now about the roles of these various hormones in the genesis of the disease. In terms of what makes the tumors, it's interesting that Bob Ney, who was a assistant professor when I was a postdoc at Vanderbilt, worked with a rat adrenocortical tumor cell line in culture and found that agents other than ACTH stimulated secretion of corticosterone by that tumor cell line. And he developed the concept of what he called promiscuous receptors, the idea that these cells, one of the reasons that they were, quotes, autonomous in terms of their secretion was because they responded to inappropriate stimulation. And a later postdoc at Vanderbilt, André Lacroix from Montreal, has elegantly extended those studies over the past several years and shown in tumors of the pituitary and of the adrenal gland all sorts of receptors that are not normally seen in these cells, and explains why they are constantly secreting ACTH or constantly secreting steroids not under appropriate control. I was actually talking to Andre and Lynette Neiman from the NIH yesterday, two good friends and collaborators and competitors, and I told them that I was having this interview today, and I said to them that I was going to say that nothing much had changed in Cushing's over the years, and Andre especially got quite exercised about that and said, well, you know, there's been a lot of changes. Now we've got new drugs. There's a new drug called pyrazinamide or something like that, and there's one I can't remember these, but and mifepristone, which was a blocker of the receptor for steroids, and saying how these were advanced. Well, we used dopaminergic agents to try and cure Cushing's back when I started with bromocriptine then. When somatostatin came along, which is what this paciricide is all about, that seemed to work in some and not in others. And very quickly, Andre and Lynette were arguing about what was effective and what wasn't and so forth. And I said, so, yeah, not much has actually changed. It's still a very interesting diagnostic and therapeutic challenge. So we know a lot more, but it's still a challenging disease.
What is ectopic ACTH syndrome, and what was the nature of your investigations in that area?
Well, ectopic ACTH syndrome is secretion of ACTH by a non-pituitary tumor. And Grant Little was the man who described that syndrome. He described it actually on the basis of ACTH bioassay in exactly 2 rats. in the Lipscomb-Nelson bioassay, and subsequently studied many, many more patients. It was the first of the ectopic hormonal syndromes. There have been many since then. It was a surprising finding. So what we were interested in doing was to investigate the peptides that were actually being secreted by these tumors. It wasn't clear at that point. Genome had not been sequenced. Numa and Nakanishi had not sequenced the bovine POMC mRNA. Eiper and Maines had not yet studied the biosynthesis of ACTH and ATT20 cells. So we were interested in whether ectopic ACTH was somehow different in nature than from the pituitary. And so we used
From the healthy pituitary?
From the healthy pituitary, exactly. And so to do that, we really needed to see ectopic ACTH secreting patients. They were fairly hard to come by. It's a very unusual disease. We needed to have tumor tissue from these patients, and that was very hard to come by in terms of getting it in a condition that was useful for us. And thirdly, we really needed to try and get a ectopic ACTH secreting tumor cell line in tissue culture, which really would give us the opportunity. And we tried many times to do that. I flew around the country trying to get tissue and starting the line in tissue culture, and were unsuccessful except in one case. We had a patient with a melanoma that was secreting ectopic ACTH, and we were able to get that in tissue culture and do some limited studies on it, but it produced so little ACTH per cell that we couldn't do any detailed analysis on it. Then finally, George Sorensen and Olive Pettengill at Dartmouth were able to get an ectopic ACTH secreting tumor in tissue culture. And using that cell line, Xavier Berdagne from Paris, who was a postdoc at the time, and others in my lab, studied the spent tissue culture medium from those cells and found all of the ACTH and MSH peptides there, as well as larger fragments of that had ACTH and immunoactivity and bioactivity. And so this seemed to indicate that the same kind of things that were going on in the ATT20 mouse pituitary tumor were going on in the human ACTH secreting ectopic tumor. We finally were able to actually sequence part of that gene and found that it was identical to the one that Nakanishi and Numa had sequenced.
Do you want to say more about how these studies advanced the field?
Well, I think that at that time it's hard to look back from the perspective of today and say how anything that you did at that time were important to the field, because we know so much more now that looking back it all seems pretty simple. But the issue at that time was whether these ectopic secreting ACTH secreting tumors were actually expressing the normal gene, whether they'd been there was some mutation or something that happened to the cell that made them secrete these peptides, what the nature of the peptides were, and so our studies were really, if you will, kind of adding bits of data from various sources that just kept building on the central theme that no matter where ACTH was produced, it was always the same kind of process. We found that it was produced in neuroendocrine cells throughout the body, for example, and so it's now pretty clearly known that in almost every case, when an ectopic ACTH secreting tumor arises, whether it arises in the lung or whether it arises in the prostate or thymus or wherever, it's actually arising from a neuroendocrine cell that has the machinery to produce peptides, and it's simply the result of the gene for ACTH being expressed inappropriately in that tissue. And the exact mutations that occur to cause that to happen in any one tissue, as far as I know, are yet not well understood, but I assume that they will be soon.
What led to your interest in epidermal growth factor?
As a medical student, I had read some papers by a Harvard anatomist named Marcus Singer. And being an anatomist, he had a viewpoint. And Marcus Singer asked the question, why is it that a tadpole can replace its tail if you cut it off, but a frog cannot replace its limb if you cut it off, the same animal? And Singer came to the conclusion that when a tadpole metamorphosed into a frog, that all of its tissues hugely increased in size except one, and that was the nervous system. So he said, well, maybe the frog has just outgrown its nervous system. So he did a very simple experiment, which I still find difficult to believe, but he took the sacral nerve from the leg of a frog, and he cut it, and he then tunneled up under the skin of the frog up into the forelimb, and he put the end of the nerve down into the forelimb, and after the thing had healed up and so forth, he then cut the forelimb off at the level where the nerve was, so he transected the nerve. And the frog regrew a new leg with an elbow and fingers and the whole shebang. And that impressed me. It still impresses me. And as I got into endocrinology, I thought, well, you know, it's got to be something the nerve is secreting that's doing this. It can't be some electrical signal. That didn't seem to make sense to me. I think in retrospect, there was a mound of cells that formed on the limb when it was going to regenerate, and I'm sure those were stem cells by what we know now. But something apparently in the nerve was directing this to regenerate. So I became interested, and I started reading the literature, and as I read the literature, because I was interested in growth, and I read growth factors, and I started reading about epidermal growth factor, and rather belatedly, in my muddled sort of way, I realized that the guy who was writing these papers was a guy named Stan Cohen, who had been a professor of mine in my first year at Vanderbilt in biochemistry, and that his laboratory was about 50 feet down the hall. So I thought, well, that's interesting, so I'll just go down and talk to Stan and see what we can put together. So I went down to Stan and I said, you know, do you have any antibodies to EGF? He said, oh yeah, we got plenty of antibodies. And I said, well, have you got any purified EGF? And he said, yeah, we got purified EGF. And I said, well, what if we try and make a radioimmunoassay? Would that be interesting? And he said, yes. Well, he was working in the mouse. The whole story is too bad we don't have time, because the story of how EGF was discovered and how or at all, and NGF were discovered were just kind of pure accidents, and how Stan came to discover mouse EGF is a classic example of serendipity and a prepared mind. But at any rate, it taken us 2 years to develop a ACTH radioimmunoassay, and it took us 2 weeks to develop a mouse EGF radioimmunoassay, and we were prepared to measure some things. So the question was, what should we measure? So the first things we measured were mouse submaxillary gland EGF. It was known that male mice had much more EGF than females, and so we first measured the influence of androgens on submaxillary gland EGF, and then it was known, and I forget exactly how that came about, but it was known that adrenergic agents, catecholamines like epinephrine and so forth, that they released EGF, so we studied the effect of EGF and the release of EGF from the submaxillary gland, looking at the blood of the mouse. Then we collaborated with Wayne Barden, who was then at Hershey, the Penn State Medical Center at Hershey, and we looked then at the effects of progestins on TFM-Y mice. These are mice that have so-called testicular feminization, and they are not responsive to androgens. So we looked at progestins, which in those mice have androgenic, synandrogenic, that means they act synergistically, and anti-androgenic effects, and studied those with Wayne. And then with my interest as always on the human, we decided to see if humans had EGF. There was every reason to believe they did. And so a postdoc of mine, Ralph Starkey, and working with Stan Cohen, looked at large volumes of human urine and purified a material from human urine that displaced labeled mouse EGF in the mouse EGF radioimmunoassay, and so we assumed this was human EGF. Shortly after that, Stan and Graham Carpenter isolated and sequenced human EGF. So with the human EGF radioimmunoassay, then we started looking at human tissues and fluids. It turns out that human EGF had been discovered, quotes and quotes, in a different way, and it was a peptide called beta-urogastrone. It turned out the two were the same. And beta-urogastrone was of interest because it blocked stimulated gastric acid secretion in the gut. So we looked at the tissue distribution of EGF in animals and in man. We found that not only in the salivary glands, but also in the lacrimal glands, the glands that produce tears, and the sweat glands that produce sweat. Found it in the duodenum and the kidney, and the thyroid gland for reasons not entirely clear to me. But it's expressed in many tissues, and its usual effect, its main effect, seems to be on the epithelial surfaces, so that it bathes the urinary tract, it bathes the eye, it bathes the gastrointestinal tract, it bathes the skin through sweat, and so when you injure any of those tissues, the epidermal growth factor stimulates its growth and regeneration. And it's why, by the way, when you get a wound, you lick it because you've got epidermal growth factor in your saliva, and that's helping you heal the wound. And dogs know it, and apparently we did too. So we had a lot of fun with that, and yeah.
What about your collaboration with Wiley Vale on corticotropin-releasing hormone?
Wiley was an old friend of mine, and he was searching, as were Roger Gilman and Andrew Shalley, for the Holy Grail, which really was for the factor that regulated ACTH secretion. They all really started out with that in the first place, and it became clear that they weren't going to do that. Unfortunately, they discovered easier releasing factors, TRH being one, which is just a tripeptide, long before. But they were looking for it, and Wiley had fractionated hundreds of thousands of sheep hypothalami like the others had done. And he thought he was on the track of CRH, and so he called me and asked me if we had an ACTH antibody in a radioimmunoassay that we could that he could use to follow the fractions. And he had basically sort of three requirements. He needed something that was reasonably sensitive because he didn't want to waste large amounts of material. He needed something that was accurate so that it actually would measure ACTH and not other things. And he needed large volumes of it because he and his laboratory were just generating enormous numbers of fractions from columns and various techniques he was using to isolate it. And it so happened that in the course of our developing the ACTH radioimmunoassay, that we had immunized lots and lots of animals, and one of the animals was a sheep, which happened to be sheep 1. And the second bleeding of sheep 1, which was bleeding 2, was a very, very good antibody. It had very, very high sensitivity, very good specificity. And we had about 1 liter of it. And so sheep 1 bleeding 2 was an ideal antibody for Wiley, and I told Wiley he could have as much of it as he needed and for as long as he needed it. So there was no quid pro quo on that. I just was glad to give it to him. We just had it sitting in the freezer. And using sheep 1, bleeding 2, Wiley was able to isolate CRH. So that's how I happened to get involved in that. And then Wiley, without prompting from me, when he isolated CRH and published the paper, contacted me and said, would I be interested in doing some studies with Hovi and CRH? And I said, oh, yes, I would. So from that time on, Wiley and his peptide synthesis guy, Jean Riviere, in the laboratory provided me with all the Hovi and CRH I needed to do the things we did, and also later provided us with human CRH. So,
and he did that just out of because he was grateful for your help?
I mean, I think the reason he made it available to me was for that reason, but Wiley was an extremely generous guy, and he would have made it available to somebody who would do the studies anyway. So I'm glad I helped him out, and I'm sure he was pleased to help me out, but I don't think it was a tit for tat kind of thing at all. I never thought so. Anyway, it was awfully nice. Mm-hmm. So we were interested in pursuing this, and the sequence for CRH, Hovi and CRH had been published, so anybody could get somebody to synthesize it or synthesize themselves. And we knew that the NIH group at that time at the National Institute of Child Health and Human Development were hot on this because they were interested in the same kind of things that I was. We were competitors and all along the line, and that group consisted of George Crusos and Lynn Lorio, Lynette Neiman, Heinrich Schulte, Phil Gold, Ed Oldfield. There were a lot of them, and they were all good. And so we knew we had limited time and needed to get going, so we put together a research team. We had 5 fellows, Rick Jackson from Australia, Rowan DeBold, DeCherney, let's see, who else was on that? Bill Sheldon and Themis Kamalaris from Greece, and myself and Don Island and Wendell Nicholson. I forgot to mention Wendell Nicholson earlier in our discussion and should have. Wendell came to my lab from Grant Little's lab fairly early in my career, early in the ACTH radioimmunoassay era, and became my man Friday in the laboratory. He was my hands, he was in many ways my brains. He was very reluctant to get into radioimmunoassay, and ultimately became one of the absolute world's experts on radioimmunoassay and how to develop them and how to do them. And interestingly enough, is still working at Vanderbilt at age 79. He and I are almost exactly the same age. For Al Powers and other peoples there doing radioimmunoassays and taking commercial radioimmunoassays that are relatively insensitive and doing the tricks that he and I learned how to do, increasing their sensitivity so they can be used in small animals and small samples of blood to study what happens when you knock out this and that in various mice and so forth. So terrific, terrific guy. So we put together a team, and the question was, what should we study first? And the first thing we had to do was to develop a dose-response curve. And there had been some troubling studies in dogs with Hovi and CRH in Wiley's lab with hypotension and so forth. And so we were extremely cautious, and we administered Hovi and CRH to normal subjects over a 30,000-fold range, starting with a 1 nanogram dose. And we checked their symptoms and signs and all the rest, and their subjective feelings, and their ACTH and cortisol after each of these doses before proceeding to the next. And we proceeded in 3-fold increments, 1 nanogram, 3 nanograms, 10 nanograms, 30 nanograms, so forth, and ran it up to 30,000 nanograms or 30 micrograms of ACTH. And what we found was that up to 1 microgram dose, things were pretty fine. But when we started getting above 1 microgram, people started having intestinal cramps, feeling not well at all, and then they developed extreme facial flushing and edema, and at the 30 microgram dose, that lasted for up to 6 hours. It was impressive and a little bit scary. And although their ACTHs continued to go up through those doses, we reached the conclusion, number one, that 1 microgram was a safe dose, and number two, that probably the doses, the responses above that were in response to stress in addition to response to CRH, and therefore were not reliable. So we established the 1 microgram dose. So having done that, well, actually while doing that, and before we published it, 2 patients with Cushing's disease were admitted to the CRC. And so we admitted the 1 microgram per kilogram, 1 microgram dose of CRH to these 2 people. And as luck would have it, one of them had a very brisk response to CRH, and the other one had a very minimal response to CRH. And so we wrote up a paper about this, a rapid communication, and I sent it to Grant Little just for his interest, nothing else. We didn't have to clear papers through the department chair at Vanderbilt. And I got this call from Grant, and Grant said, "You can't publish this." And I said, "What do you mean I can't publish it?" He said, "Well," he said, "you know, one of these people basically didn't respond, and the other one did respond." He said, "It doesn't make any sense." And I said, "No, I think it does make sense. It means that some people with Cushing's disease don't respond very well, and some do, and it's probably going to turn out to be an unreliable test for Cushing's disease, and people ought to know about that." And anyway, it turned out that's exactly the case, that using responses to peripherally administered CRH was not a useful test to diagnose Cushing's or to differentiate Cushing's disease from ectopic ACTH syndrome. So then we went on to look at diurnal rhythm, possible diurnal rhythm response to CRH, to look at the effects of the ambient level of cortisol when you administer the CRH, to look at the effect of eliminating their cortisol response. We gave patients, normal subjects, huge doses of metirapone to block their adrenal from making any cortisol, to see what happened. And the interesting thing with all those things, it turned out that cortisol had almost no effect on whatever's level, no effect on the first 60-minute response to CRH, but had a huge level on the subsequent. The response was biphasic, and the subsequent response was greatly influenced by cortisol, which seemed to suggest that the initial response was all or none, but then the actual cortisol response to CRH itself then fed back on the pituitary and modulated the subsequent continuing response to CRH. We looked at the response of CRH in combination with insulin-induced hyperglycemia, which is a stress response. We looked at it in combination with AVP, arginine vasopressin, which is another releaser of ACTH, and published all those kind of papers. Then my research was always kind of equally balanced between the bedside and the bench, and that's sometimes a good thing and sometimes a bad thing. If it's a good thing, then the basic researchers think of you as a basic researcher, and the clinical investigators think of you as a clinical investigator. If it's a bad thing, then the basic researchers think of you as a clinical investigator, and the clinical investigators think of you as a basic researcher. I'm not sure how I come out on that spectrum, but anyway, that's what I had fun doing, and that's what we did. So while we were doing these things in patients, we also developed radioimmunoassays for ovine CRH and then human CRH, and looked at plasma disappearance rates for those 2 hormones in humans. We were also working in a microperifusion system that we developed, which was a tiny little column, 50 microliters, little capillary tube basically, in which we loaded normal pituitary cells from rats. And then we infused them with CRH alone, CRH and AVP alone, and angiotensin II and oxytocin, all things that had effect on ACTH release, and then did them in combination. And we were able to follow ACTH secretion in that situation on a second-by-second basis. Toru Watanabe, another of my many Japanese postdocs, did that. and we found that ACTH was released within 5 seconds of the time that they saw CRH, and that CRH, in the absence of any cortisol negative feedback, which there was not in the system, continued to be secreted for as long as we infused ACTH, so that the CRH was apparently stimulating synthesis of ACTH, of POMC, as well as stimulating secretion. So it's basically a limitless system as far as we could see. In contrast, the other agents, AVP, oxytocin, and angiotensin II, induced a very rapid response also to, in terms of ACTH release, but that spiked very quickly and then disappeared. So even though you continued to infuse these, they had no effect on the pituitary cells. And when you gave them in combination, they were synergistic. You got a much bigger response than to CRH alone, but that effect also was short-lived and disappeared and went back to the basic CRH situation. So I think we were able to demonstrate in very great detail how these things worked at the cellular level, and they were reflective of what we saw in clinical studies in humans.
Now, I know you've discussed the Endocrine Society in pretty good detail with Dr. Friedman. I guess that was 2002, I think.
2003.
But I'd like to ask you a little bit more about the Endocrine Society.
Sure.
There could be some overlap in what you already said. Would you talk a little bit about what the Endocrine Society was like when you first became a member in the mid-'60s and early '80s?
Well, of course, the Endocrine Society to me was a wondrous thing with giants in the field, the members of it. But it was, in fact, a very small society. I remember the first annual meeting that I went to was in Chicago, and Nicholas Christie, who later became a colleague and friend, chaired my session at that meeting. And the entire program for that meeting, I think, was something like, I don't know, 30 pages long. It was a little pamphlet, basically, and compared to what's going on here this weekend, I mean, it just was no comparison. It had been formed in, I think, 1916, as I recall, by a group of academic endocrinologists. They called it, I think, the Society for the Study of Internal Secretions. It was later changed to the Endocrine Society. It was at a time when endocrinologists were thought of kind of as quacks in the medical community, because they were mainly administering extracts of various glands into people for diseases, and it turned out that most of them didn't contain any hormones. The gonads and the adrenals contained very, very little steroid hormone. The one that really worked and sort of made things go was the thyroid, which contained a significant amount of thyroxine. Matter of fact, they were still giving thyroid extract when I was a postdoc. So it was a society that was basically academically oriented. It was an old boy kind of organization that had fairly strict requirements for admission. It had was publishing 2 journals when I joined it, the JCNM and Endocrinology. It was putting on this annual meeting and was providing otherwise very few services to its members, and had, I thought, kind of limited scope as to what it could do and where it was going. But I was very glad to be a member. By the time I got involved in the society, it had progressed somewhat, but it was still very, very small. The central office consisted of Nettie Karpin, who was a wonderful Jewish lady of indeterminate age, probably in her 70s at that point. She was a second mother to us all, a marvelous woman. I think my wife still considers her a second mother. She had 2 full-time people who were with her. They occupied a tiny, tiny little office suite that we rented from FASEB on the NIH campus. And that was kind of about it. Bert O'Malley was president of the society, and Bert called me up and asked me to be a member of the finance committee. I told Bert I had absolutely no expertise in finance whatsoever. Bert said, "Don't worry about it." He said Nettie and Nick Christie, who was the secretary-treasurer by that time, would, you know, cover my back, and you'll have a good time. So I said,
"This was your first time on a
committee. Yeah, it was my first connection with the Endocrine Society and other than just as a member.
And this is around the early 1980s?
Yeah, I forget exactly where it was, but yeah, right about there. And Al Wilhelmi was the chair of the committee, and they had Al had very wisely set up a sort of a rainy day fund of money to carry the society, particularly its publications of its journals, if something inauspicious should happen. And that was managed by a group called Shields Assets Management, which was a company that dealt primarily with nonprofit and mainly academic institutions. So they had our money invested, and the society was in good financial shape. We were making money from our journals, primarily from our membership dues and from the registration fees for the annual meeting. The problem was.
What about expenditures? What were your expenditures?
Well, the expenditures were fairly simple. I mean, the expenditures were the cost of putting on the annual meeting and the costs of putting out the journals and the central office staff. There was really very little being done in terms of outreach in any other way. No lobbying that I knew in government, no nothing. So it was limited income, limited expenditures, and limited kind of scope of where they wanted to go. And I have to say, everybody was, I think most people were very happy with that at that time. There were relatively few endocrinologists in the world. Most of them were in the society. The people in the society were not interested in bringing clinical endocrinologists into the society. That was a totally different game. These were really hardcore academic people, most of them basic, I guess, and some of them clinical researchers like Grant Little and so forth. But they were certainly not interested in the business of endocrinology, to put it that way.
Then things took a big change.
Then things took a change. I became chair of the finance committee, and that was fine. And then there were some rumblings that were going on near the end of my tour on the finance committee. A guy from the West Coast, Al Parlow, who was a very entrepreneurial sort of guy and was at that time the director of the National Pituitary Agency, which was an organization was associated with the NIH and with the Endocrine Society, which provided all of the pituitary hormones and the human pituitaries that were collected and so forth. It had been founded by Al Wilhelmi, who was a growth hormone expert, and they needed some sort of group to take care of the human pituitaries that they collected, which were then used to extract human growth hormone, which was used to treat children with growth hormone deficiency. And so that's how it started. But it then evolved into a much bigger kind of organization that stored all sorts of hormones and tissues and so forth and so on. And so Al was the head of that at that time, and Al sort of did the unthinkable. He challenged Nick Christie for the office of Secretary-Treasurer. And Al wanted to bring the society into the 20th century. He wanted to put things on computers. He wanted to just hugely expand and also centralize, because Al was a big hierarchical sort of guy, centralize things in the central office. And the older members of the society were suspicious of change. They were suspicious of administrators because in their institutions, I think they found in general that administrators were hindering them rather than helping them in those days. And they were not excited about this thing. And Parlow was turned back. He didn't successfully challenge Nick, but it was a symptom of what they call in the business the young Turks in the society of getting restless and wanting to move on. So they asked me to run as Secretary-Treasurer. Nettie and Nick convinced me that it would take less time for me to be Secretary-Treasurer than it was as Finance Committee Chair. That turned out to be a gross misstatement of the facts, at least as I was Secretary-Treasurer. And I said, well, I would do that, but I would do that only on one condition, and that is that there actually be an election, that there had to be, council had to come up with another candidate for the members, because I just was not happy with this perception of the old boy Neckward of sort of nominating themselves and their buddies and so forth. And so they said, okay, and I honestly forget who ran against me, and I don't know anything about the nature of the outcome other than the fact that I got more votes. How many more votes, I have no idea. So I became Secretary Treasurer-elect for a year, and while Nick was still Treasurer. And while that was going on, Nettie Carpenter announced that she wanted to retire. And it was time for a retire, I think. And so Nick Christie and the then President, Jerry Auerbach, and myself were appointed by council as a committee to find a new executive director. And we got a lot of advice from the old guys in the society, the old presidents and mainly. And they thought what we ought to do was find an ex-president of the society, retired endocrinologist, to become the executive director. Well, all three of us had different ideas about that. We wanted to move the society to a more professional kind of basis, and in particular, we wanted to get publications in-house rather than being farmed out. Reason for that, do you want to go into,
yes,
go into that? Well, the reason for that was because the Publications Committee worked with Williams & Wilkins, who was our publisher at that time, and the Publications Committee negotiated contracts of usually, I think, 3 years of time with Williams & Wilkins, and they had a very comfortable kind of relation with Williams & Wilkins. Secondly, because the major source of income for the society was this subscriptions from publications, the Publications Committee developed a very proprietary kind of feeling about the whole thing. So they felt that since they made most of the money for the society, they should also have a major say in how the society was run and how that money was spent and so forth. Well, it turns out that the contracts that they negotiated with Williams & Wilkins were not that good, despite the feeling that Williams & Wilkins was terribly collegial about this, because they contracted for a given number of pages for that period of time. And any pages over that, the society bought page by page at a much higher rate. And there was a conflict of interest built into this because the Pubs Committee worked with the editors-in-chief of the various journals. The editors-in-chief were all entrepreneurial kind of guys, and they wanted to publish as much good science as they could. Endocrinology was expanding by leaps and bounds, so over the 3-year period of the contract, they were getting more and more manuscripts being submitted. So to maintain the same acceptance rate, the percentage of the things, they had to publish more and more pages. So by the end of the 3 years, we were publishing far more pages than we had contracted to do 3 years before and paying through the nose to publish them. The Publications Committee felt that they needed to let their editors-in-chief do what they could do. They were putting in enormous amounts of time. They were interested in the society, et cetera. So there was this kind of impossible kind of conflict of interest. So what we wanted to do was take publications into the central office where all of those kinds of things would be within the society so that the society could make judgments on how many pages. We wouldn't have unexpected changes in paper and cost of paper and so forth and so on. And getting the Publications Committee to agree to that turned out to be a very difficult thing.
But it was key.
It was key. And so in the process of looking for a new executive director...
And finding a new director was the other key.
Finding the new director was the key, because we had to find somebody who had expertise in doing that. And so we identified 3 people, all of whom had been executive directors of other societies. 2 of whom were, in I think my opinion and the opinion of the others, kind of doing this as kind of an end sort of deal. They'd already done their thing with a big society, and this was a small society, and they could sort of phase out. That's not what we wanted. And Scott Hunt was coming from a relatively small society of immunologists. He had all the criteria. He and his director of publications with immunologists had taken publication in-house without any difficulty, same kind of situation we had had. And so we were absolutely sure that Scott was the guy we should have, and he's the guy we selected, and council approved it, and so that was that. So then the problem was, where do we go from there? And Scott had a hard time because he was regarded with suspicion by lots of the old guard. He was certainly regarded with suspicion by the Publications Committee when they got wind of the fact that we were thinking of taking publications in-house. And Publications had been asked about this 2 or 3 times before by council to study it, and they always found very good reasons we shouldn't do it. So kind of the stage was set for a big problem. About the same time, Bert O'Malley and his friends here in Houston proposed we start a new journal, Molecular Endocrinology. And because endocrinology research was moving in the molecular biology direction, and they felt that we should do this. But from our experience with previous journals, we knew that we were going to have 3 or 4 years where we were going to putting a lot of money out and having very little come in from this sort of thing. And so that was another issue that was going on. The Publications Committee did not want to have that journal start. So finally, we had a meeting kind of bringing these things to a head at Georgetown University in Washington. And that's where Scott and I had the famous, at least those who knew about it, walk in the park. And what happened was that the then director of publications, after hearing all the arguments and so forth, came up with a brilliant proposal. And he suggested that we test the system by taking molecular endocrinology and publishing that in-house and seeing how successful that was in terms of generating funds and so forth. Well, we knew already that it was going to lose money for 4 years. There was no way it was going to succeed in any kind of financial kind of sense. So basically, he was sinking both in-house publishing and molecular endocrinology with one fell swoop. And I had had some heart problems. I'd had a bypass surgery prior to that time, and I thought I was going to start having angina. I was really, really upset. I was looking across at Scott, and I thought Scott was going to have a stroke and or at least resign from the society from within the week. So I grabbed Scott. We called a recess, and I grabbed Scott, and we went downstairs, and there was a athletic field and a track down there, and I think Scott went around, and I went around that track, sort of my hand on his shoulder for about 2 or 3 times, calming him down, and he was really, really upset. He felt, and I think quite rightly, he felt attacked and insulted by all of this, and so it was rough. But to make a long story short, we brought Lenny Miller over from immunology. Lenny and Scott moved everything in-house. There was never a ripple. Our publications income went sky high as the result of not paying profit to a middleman, and everything worked out fine.
More about the vision you had for the society, the reorganization you imagined?
The problem the society had, in my view from my experience of going to council meetings and so forth, was that the council, when they came to meetings, was not well prepared. It wasn't their fault. They just didn't have an agenda set up. They didn't have background information provided soon enough. And they didn't have the resources in the central office to run the society in a way that, with which they were entirely comfortable. So what happened was that council came to these meetings having read what material they had on the airplane coming to the meeting, which is the way we sometimes do lots of things. And I found council talking about the same things over and over, year after year, meeting after meeting, number one. Number two, they had a very hard time reaching decisions. Number three, they micromanaged the society. I mean, everything that the central office wanted to do, everything that the publications committee wanted to do, everything, they had to be involved in every single decision. When Scott, well, as one of the reasons for getting Scott was an MBA. He had experience in business. He knew how to run an office. He knew how to run a society. And my goal was to get council to get away from micromanaging the society and let Scott and the central office do that, let them do their thing, and have council develop policy. They were the ones who had the scientific purview. They were the ones who were looking down the pike from the standpoint of where endocrinology was going, which Scott didn't pretend to have. And let Scott then provide the means and the mechanisms and the facilities and the money for them to do that. So the first thing was to get council to accept Scott. And that took a while, because it was the same thing. They were suspicious of administrators, and they questioned Scott at every turn. And every time Scott took on some new activity which he had suggested or which council had suggested to him, Scott needed more people. I mean, he couldn't do everything. Well, every time he requested people, they say, well, you know, we gave you 2 positions 6 months ago. Why do we need 2 more now? And he would have to explain it to him. So it took a long time to do that. Then the second thing was that even in terms of the scientific aspects of the society, council met twice a year. And they met for a day. But that was not enough to run the day-to-day scientific kind of aspect of what was going on in the society. So I felt we needed to have a subgroup from council to deal with that kind of thing. So I suggested that we have an executive committee. And I suggested that the executive committee consist of the elected officers of the committee, Scott is an ex officio member, and 2 members of council to make sure that council was comfortable with the fact that somebody wasn't running away with their show. And surprisingly, I still don't know quite why. Council was quite happy not having council members on it, which I guess was an expression of their confidence in the officers. So that's what we did. And the way we did it was using email and so forth. We sent out agenda and background material and so forth to members of the executive committee, and we met every month by conference call. We didn't want to add more travel to our already busy schedules, and it turned out that the conference calls worked extremely well. And so we made decisions on a month-by-month basis throughout the year and reported those in detail to council. I think council never had any problems with any of them. So that moved that enabled the society to move much faster than it had before. It moved at a kind of a glacial pace before, and now we were beginning to pick up speed.
Now, these changes were supported by were there factions that wanted different changes?
To a degree. For example, there were people, the chair of the publications committee, at least one of the past presidents of the society who came to me and said, you know, we shouldn't be doing all these things. That's not what we're about. We're an academic, scientific society, and our goal should be to spread the scientific word as well as we can through our publications and our annual meeting, and that's it. And the way we should do with our money that we get is to turn it back to the members and make their membership and their journal subscriptions and their registration fees at the annual meeting as small as possible. Some of them were actually unhappy that we even had a reserve fund. They just thought that was unnecessary, and fortunately Al Wilhelmi had convinced them at least we needed a little one. It wasn't adequate, but we had a little one. And the other thing was that there was this odd feeling at the head of the society that we shouldn't burden, I guess that would be the term they might use, the members with the financial affairs of the society. So I remember, I guess it was the meeting when I was the Secretary-Treasurer-elect, I don't know, but it was one of the last meetings, annual meetings, that Nick Christie was Secretary-Treasurer, and he was giving the Treasurer's report. And he had this report which had been made out by Nettie Carpin, and you know, it was like 3 pages or something like that. And Nick started to do this thing, and he took it and threw it over his shoulder onto the stage floor and said, "Oh, you don't want to hear all of that." He said, "The society's in good shape. You know, we're doing just fine." And that was it. And I was very opposed to that also. I thought that there ought to be absolute transparency of what the society was doing financially, and so when I was Secretary-Treasurer, they got a full report, and they got a written report if they wanted it. And so it just, and again, Scott was totally with that. I mean, he was for a really professional kind of operation at the level of the office. The young people were fine. Council, after a very short period of time, they were fine. There were issues as to how much money and how much resources the society should put into this effort or that effort and so forth. But the more and more Scott was given his reign, the better it got. I mean, for instance, here, the money that we're getting from exhibitors, tremendous income. Early on, that was not the case. Advertising in the journals. There were a lot of guys in the society when I was Secretary-Treasurer who wouldn't hear of having advertising in the journals. That was dirty business. They didn't want to get involved in that. Well, later on, when ACE was about to form, the big concern was that we'd lose our advertising to their journal and lose our exhibitors to their show. So things just changed. And I think the other thing that changed was that academic medicine became less of an ivory tower. There were more and more people who were involved for a variety of very good reasons with pharmaceutical companies and so forth, and consulting deals, and actually forming their own companies. And so the interaction of business and the academic science became more and more accepted, and that brought its problems. I mean, then there was the question of ethical conflict of interest sort of situations the society had to address.
So, in your mind, the society didn't really have the option of staying like it was. It was going to have to, something was going to have to change. Well, first of all, I belong to a number of societies, some of which, one of which I resigned from, although it's a very auspicious society, because they refused to admit as a member somebody that I felt absolutely sure deserved being a member. And it was because, in part, because they had a very limited number of members they would take in. They take in 25 a year. And he just didn't fit in that 25. And I wrote them and said, if it ain't good enough for him, it ain't good enough for me. And P.S., my opinion is that if you maintain this kind of exclusivity, you're going to lose your relevance. It's just not going to last. So, you know, good luck to you, but I'm out of here. And that was your concern for the Endocrine Society?
Yeah, I think I have two tongue-in-cheek laws. Orth's first law is big is bad. or second law is small is not necessarily good. I also have the, there's no free lunch rule. So you can't, you don't get something for nothing. So when you get big, you lose a lot. I mean, I used to know virtually everybody in the society. I walk around here now, I don't know anybody. So that's a problem. And then communications becomes a problem. So how do you tie these people together? How do you keep a coherent whole? How do you have some sense of purpose, especially when you've got a society like ours whose biggest asset and biggest problem is the fact that it has three very, very distinct constituencies, practicing endocrinologists, clinical researchers, and basic researchers. So I think the society has done and continues to do a great deal to address those problems, and has done a good job of it. But I think that if we had stayed at the size we were back when I joined, I mean, some other people who are neuroscientists would have been in the Neuroscience Society, and people who are this, that, and the other would have been in some other society. They wouldn't be here. And our advantage is that because we're so diverse, we bring these people to one place where they can talk to one another.
As president of the society, and then past president and immediate past president, 1996 to 1999, what were the most compelling issues you faced, and what were your goals?
Communication was a major one. The various committees were perennially upset with council because they didn't get initiatives that they proposed to council funded and approved. And the issue was because the society had limited funds, and somebody had to decide where they went. So my idea was to bring the committees together in such a way that they would know what the other committees were doing, what council wanted them to do, and it would give them, might not make them entirely happy, but it would at least give them an appreciation for, where they were standing in line and why they didn't get chosen this year. So communications between members of council and committees, between committee chairs, and between members of the committee was, I think, a big thing. I had a show and tell session for the council meeting once a year where everybody got up and said what their committee was doing, what they'd done that last year, accomplished, and what they hoped to accomplish the next year, and so forth. And they all listened to it. I don't know whether they've continued that or not. It made for a rather long meeting, but it served its purpose. Another thing that I was concerned about was the growth of the society. When I joined the society, its governance, we were a society of around something less than 3,000 members. And we had an annual cash flow of a little over $1 million. And I was told yesterday that the cash flow is now 42 million, something like that.
I don't know.
And there are twice as many people at this meeting as there were members of the society when I joined it. Amazing. I mean, just amazing. And so the question was, well, where are the members going to come from? And so we wanted to attract clinical endocrinologists, and the society did that in part by broadening their criteria for membership so that clinical endocrinologists could be members in good standing. I saw endocrinology as an international enterprise, and there was a society which my mentor, Grant Little, certainly helped to establish, the International Endocrine Society. But I had served on the central committee of that society and helped plan one of the meetings, and I was impressed with the fact, at that time at least, that there was a great deal of sort of global politics in that. So the people who got to present at that meeting weren't necessarily the best people. They were the people who were from this country and that country and this part of the world and so forth, which was fine, but it wasn't the best science. And I thought that they, in general, would be a lot better off if they came to our annual meeting. And so I wanted to increase the international membership. And we did that. And to do that, I felt that we had to offer the international members full full membership. So that meant we had to have people who would be officers of the society, who were on council, people who chaired sessions, and people who got the awards of the society. And I think that's happened, and I think I helped to start that, and I'm pleased about that. I mean, I look at the list of presenters and where they're from at this meeting. It's everywhere there's an endocrinologist, they're here. It's remarkable. And then I was talking to Lynette Neiman and Andre Lacroix yesterday, both of whom are on the International Endocrine Committee, Central Committee, which, like I was. And I like what's happening there. They're changing the focus of the International Society, apparently, to fostering endocrinology and supportive endocrinologists in undeveloped sections of the world. That's good. That's great. Third thing I was concerned about was ethics. I had become aware of several ethical problems in other academic societies, and we had an interesting episode in ours when Maria New, who was president at the time, tried to bring together a Congress to talk about or a symposium to talk about growth hormone administration in children with short stature. And she was reacting to, I think, an industry-sponsored symposium in which they had pushed this for very obvious reasons. But when Maria tried to put this symposium together, she couldn't find hardly anyone who was an expert in growth hormone who wasn't in some way financially involved with a pharmaceutical company producing growth hormone. So that's okay. I mean, that I don't find any problem with that, as long as people know where you're coming from. So that means you need to have absolute disclosure. If you have that, then you can let people judge whether they want to take your opinion or not. And so I asked Stan Korman, who had talked to me a year or two before, I guess, about an ethics committee and that he'd like to be involved in one. I asked him if he would be the chair, and he said yes, he'd love to. And then he named a couple of people, and I knew of another person, Paul Komisaroff from Australia. So I got to deal with both my international membership ideas and ethics committee in one, and Paul served for 3 years, I think. He's got that started. And then I had been involved in the Endocrine Society getting into Up-to-Date in Medicine, which is this CD-ROM now online textbook of medicine. I had recommended that the society do this about 6 years before they actually got involved with Up-to-Date. And the Publications Committee had lots of other things on its plate for that period of time. But when Bill Rosner became Pub's chair, he resurrected this thing. And another one of those things of serendipity, Bud Rose, who was head of Up-to-Date, he was a nephrologist from Harvard, he came to the meeting with a little exhibitor booth. So Rosner saw this booth and went over and talked to him, and Bud and his IT guru had put together basically exactly what I'd proposed. I didn't know how to do it, I just had the concept. And so we piggybacked on Up-to-Date, and I wrote and edited the adrenal section for 6 or 7 years before I decided it was time for me to bow out of that. And so we were the first of the subspecialties other than nephrology, and now all of the medical subspecialties and pediatric subspecialties are all represented on Up-to-Date. Well, the reason that was important was because the subspecialty board examinations were given by the American College of Physicians, and I'd also served on one of those, Medical Knowledge Assessment 4, MECSEP 4. And I was not terribly happy with the way that process went, and others who had participated or been involved since that time were not terribly happy, and I said, let's bring it in-house. And so Bob Kreisberg at Birmingham offered to be on that committee, and we had the advantage that we already had a syllabus which was up-to-date in endocrinology. So we used that as a syllabus, and Bob and his committee worked like dogs for, I guess, 6 months, putting together the questions, which is not easy to do, and put the whole thing together. And so we got to announce that at my meeting when I was president. So that was terrific.
one final question. What are your current views of the field?
Oh, I think it's incredibly exciting. It's, I look back on, on what I thought about endocrinology when I was a medical student, and believe it or not, when I was a medical student, they did not yet know whether human beings had 46 or 48 chromosomes. I mean, what has happened in that period of time? We now not only know how many chromosomes, we know every nucleotide in the DNA and those things, and yet there's just vast amounts that just keep coming out every day. It's impossible to keep up. Wonderfully exciting. The field itself, from kind of a practice and practical interest standpoint, has veered dramatically over the last few years because of the obesity epidemic and diabetes. So I was talking with Al Powers, our division chair, yesterday, and it's just very hard to get grants funded in anything except diabetes and metabolism. So I think, you know, there are going to be challenges there in terms of maintaining the forward thrust in adrenal and thyroid and parathyroid and all that. But all in all, I think it's incredibly exciting. It's different than when I came into it, but it was different for me than it was for the guys my age when I came into it. So I just think it's great, and I think that the other wonderful thing that's happened, which endocrinology somehow needs to be proud of, is that most of the techniques that we developed, ways of looking at things, have now been taken over by nephrology and cardiology and gastroenterology and even neurology. And so endocrinology was the rational discipline in medicine when I started, and it's now sort of spread that rationality in all directions, which is a most satisfying sort of thing.
Thank you.
Thank you.